So nice question now on nerve impulses, and explain the role of myelin. Well, you remember what happens. We've got our axon of the nerve there, and the myelin is the Schwann cell that is wrapped around this, and the nerve impulse will jump from node to node. And so let's say what myelin does. The function of it makes the nerve impulse move faster, makes the nerve impulse move faster. What does it do? It provides electrical insulation. So electrical insulation. So we only get action potentials generated at these nodes of Ranvier. So action potentials or depolarization only at the nodes. We get saltatory conduction, or that is, jumps from node to node. This also prevents the loss of lots of ions, maintains a gradient, but that's really, there's 2 marks, and those are the main things you're going to say. Bit of graph reading skills. Give the change in membrane potential between points A and B. So A, that's A and that's B. So A and B goes from -70 to +30. They've been a little bit naughty here with having a graph that doesn't have 0 on it. So our 0 would be there. So 70 from, that's 70, and then that's 30 there. So the change is both of those, so it's 100 adding them together. So 100, and I always give the unit, there's the units there, millivolts. And then a chunky bit for 6 marks. Explain the changes in membrane potential between A, B and C on the graph with references to the membrane permeability of sodium potassium ion. So a classic bit of biology. So really what we're talking about is this part of the graph being resting potential. Then we've got between A and B, this is depolarization. So that bit there is depolarization. And then over here we've got repolarization. So repolarization. So that bit. Then down here, this might be when it becomes hyperpolarized. So hyperpolarization. and then you're going to be back to resting potential again. So we just need to talk about those things. So let's start off talking about resting potential. So we're going to have, let's mention a number, minus 70 millivolts. What's happening? We have got our sodium potassium pump. So our sodium potassium pump. So 3 sodiums out, 2 potassiums in. And that means we're going to generate a concentration gradient for sodium outside. So we've got creates a Na concentration gradient. The inside is negative compared to the outside. Inside of the axon, negative compared with the outside. So that's resting potential. And if you had a diagram, your diagram would show, so resting potential, you would have sodium being pumped out across the membrane, potassium being pumped in. But because there's 3 out and 2 in, that means the inside is now negative in comparison, so it's minus 70. We could say this is now polarized. And then the next we're going to talk about, so that was resting potential, which was at A. Then it wants A to B, what's happening there. And that is our depolarization. Depolarization. And so depolarization, we've got our membrane and we've got lots of sodiums outside, and our sodium channels are going to open and sodium is going to rush in, diffusing very fastly. So we just need to remember, you only need 6 of these marks. So depolarization, sodium channels open. So sodium ions entered by diffusion. Sodium ions enter the axon by diffusion. Our membrane potential goes from minus 70 millivolts to plus, on this one, plus 30. Assuming that a threshold is met. This assumes a threshold is met. And I'm also going to mention it's an all or nothing response. And eventually these channels are gonna close. So next we are going to say that, you know, between B and C, what's going to happen. So B and C, and we're going to get repolarization. So we're going to get from plus 30 millivolts to minus 90 millivolts to start with. So what can we say about the sodium channels? Well, these voltage gated sodium channels, they are going to close. So sodium channels close, but potassium ions will start moving because potassium can now move. So repolarization. We've got a lot of potassiums in here, and these have channels as well, and these can start moving out. So potassium channels open. Potassium ions leave the axon by diffusion. So potassium ions leave the axon by diffusion, and that's our repolarization. We can say there might be hyperpolarization. Maybe in this example to minus 90 millivolts, and that's sometimes called the refractory period. But that's good because it means that we can't get another impulse just yet. So we can't, you know, reach the threshold. So another impulse is impossible as threshold cannot be met. And then we re-establish our normal resting potential. So resting potential, can't spell it, resting potential re-established at point C. And that's because of the back to our starting position of a sodium potassium pump running with all the channels closed. So K plus channels close, sodium potassium pump starts. And we're back to our starting position now. And you just needed 6 of those things in the right order, making sense to get your marks.